All right, Alessandro, we'll go ahead and have you take it away.
All right. Thank you, Mitchell. And well, hello, everybody again. Thank you for coming and for sticking around for the last session. We have three fantastic talks lined up. Those are going to be right after my talk. So please bear with me for the next 15 minutes. Hopefully, I can tell you something interesting, but the big bang is coming after me, I think. Well, I'll assume that most, if not all of you, will be familiar with the concept exemplified by this slide. And basically, the idea is that biological aging is a core mechanism that is underlying the onset and progression of many chronic pathologies that have higher incidence with age, like diabetes, cancer, heart disease, Alzheimer's. We've heard about some of these in the previous talks. Now, one obvious corollary of this idea is the so-called Geroscience hypothesis, which states that if we were to understand biological aging and somehow treat it, we could prevent the onset or mitigate the severity of all of these age-related diseases. Now, today I want to talk to you about something a little bit different. And what I think we are investigating or exploring now is a new aspect of geroscience and how geroscience can be actually applied to what are, at least at first sight, at first glance, some age-independent diseases, and how this approach can actually tell us something new about the biology of aging. So in the lab that I'm working in, we've been approaching this idea on two fronts. And in the interest of time today, I'm not going to touch on upon our work on mitochondrial disease. But if you're coming to the GSA meeting in a couple of weeks, I'll have a poster there depicting some of the geroscience approaches to mitochondrial disease. Or else I'm going to talk to you about this side project that I started around 2 years ago that I hope to turn into a full-fledged research program exploring how geroscience can help us understand and treat obesity. And the reason for obesity, well, obesity is quite a problem. It's a growing health epidemic. Obesity is defined as having a body mass index over 30. It's a growing health epidemic, as I was saying, especially here in the United States, where the incidence of obesity has been rising steadily since the 80s. And it's now attested at around 40% of the adult population that is obese or morbidly obese. And if you look here on the right at this map from the CDC, the problem is worse in some states as opposed to others, but the fact that there's very little green on this map is actually concerning. And the reason for that is because obesity is a great risk factor, and I think Crystal touched upon it earlier, and Anna too. Obesity is a great major risk factor for a lot of chronic pathologies. And one of them, for example, is metabolic syndrome, which is a cluster of conditions that make you more susceptible to cardiovascular diseases, which are the #1 killer in the United States and other Western countries. And some of these conditions are visceral obesity, atherogenic dyslipidemia. So when you have low HDL cholesterol and high triglycerides in your blood, insulin resistance and hypertension. Now, the interesting fact is that another major risk for metabolic syndrome is, you guessed it, age. The older you get, the more likely you are to have metabolic syndrome. And the parallels between aging and obesity actually do not stop there. You may have noticed we are in the midst of a global pandemic of this new disease called COVID-19. And you probably all know that COVID-19 becomes more and more fatal the older you are. But again, being obese is a pretty big risk factor for COVID-19 and for having a severe a severe case of COVID-19. In this publication here, but to show that people with a BMI over 30 or even over 35, so obese and morbidly obese, are more likely to get COVID-19 and severe cases that require hospitalization and even be hooked up to a ventilator in the ICU. Also, we know that there are interventions that are effective for both conditions. We touched about on that earlier in the symposium, but yes, the gold standard in aging research is caloric restriction, which is in a way a form of dieting, which is the first line of addressing obesity. Exercise is a good way of trying to stay healthy, also later in life. And again, it's also on the front line of treatment of obesity. And if we look at pharmacological interventions, some of the most prominent pharmacological interventions in geroscience, like acarbose, for example, or metformin, are actually anti-diabetic drugs. They come from the diabetes field, which is probably the most common complication of obesity. So what about everybody's favorite longevity intervention, rapamycin? Now, there are some conflicting data on the effects of rapamycin on obesity, and I'm not gonna go into details about them here in the interest of time, but let's just say that the question remains open. Does rapamycin prevent diet-induced obesity? And so we tried to answer this question with a small side project where we got these were 8-9-month-old black 6 mice from Charles Rivers, male and females, and we had to put them on a high-fat diet received with 60% kilocalories from fat, or a sucrose-matched low-fat diet with only 10% kilocalories from fat. And these mice were either untreated or they were given 8 milligrams per kilogram of rapamycin via intraperitoneal injections every day. And this is a concentration, this is a dose of rapamycin that achieves serum concentrations that are roughly equivalent to 27 times higher what you get with the dose that was given in the initial ITP study showing that rapamycin extends lifespan in mouse. So what did we get out of this study? The results were actually pretty good. If you look here, this is on the left, it's plotting a percent weight change. And we have a low-fat diet, there's not much happening, a little bit of weight loss with rapamycin. And on the high-fat diet, the mice being fed the high-fat diet and being on the control treatment got nice and plumpy and heavy, whereas the mice receiving rapamycin with the high-fat diet actually they did not gain that much weight at all. And they were pretty much in the same ballpark as the low-fat diet mice. Then if you look at the body composition, we looked at the fat mass via quantitative MRI. The result is similar, where you don't have much of a change in the low-fat diet mice. The mice fed the high-fat diet become nice and fat, and when you give them rapamycin, you can blunt this effect. So we were trying to figure out possible mechanisms, and we stumbled upon some publications from the Kalkoven group at the ERIBA consortium in Groningen in the Netherlands. And they were describing this transcription factor, CBP beta, that was was working downstream of mTORC1 signaling. And I'm going to spend a minute talking to you about how this transcription factor works, because it's kind of important going forward in the talk. The CBP beta gene produces an mRNA that has 4 open reading frames. These are these LAP1, LAP2, LIP, and this small upstream open reading frame. And they translate 4 different products. LIP, which is acronym for liver inhibitory protein, is only the C terminus of this gene, of this mRNA, and basically only has the DNA binding domain of this transcription factor. Whereas LAP2 and LAP1 encode for the full length protein, and they have there is both the DNA binding domain and the transactivation domain. So this is a full-fledged activating transcription factor. In fact, LAP stands for liver activating protein. Now, LAP1 is not in a particularly good context for translation. It gets translated, but very weakly. LAP2 is and LIP are instead in a pretty good context for translation initiation. But LAP2 sits right in 3 terminus of this upstream ORF, open reading frame. And usually in conditions where mTORC1 is activated, what you have is the ribosome will translate the UORF peptide, and then it will skip past LAP and translate LIP. If instead you inhibit mTOR signaling with rapamycin, and you inhibit ribosomal dependent, if you inhibit ribosomal translation, what happens is that you tend not to translate this UORF as much, and you end up translating LAP instead of LIP, as you can see here in the Western blots. And this has the net effect of increasing the LAP over LIP ratio. And in this paper here, they show how doing that actually has a lot of beneficial effects, which I'm going to show you in the next slide. Now, the Kalkoven group engineered a mouse where they deleted this upstream open reading frame. And this mouse essentially doesn't make any LIP, and it only has LAP. What they were seeing with this mouse were very interesting effects, because unopposed expression of CBP with a lap actually increased lipid oxidation, reduced lipogenesis, and also the size of white adipose tissue. Here via our TPCR, they were looking at the expression of different genes involved in bed oxidation, and here in the maroon were the delta UORF mice, and the expression of all of these genes involved in bed oxidation is increased. Lipogenesis is reduced. Looking at total fat, these mice are leaner. They have less visceral fat and less subcutaneous fat. Not much of a change in lean body mass, and there is a little bit of white adipose tissue hyperplasia, because you have more adipocytes, but they're all of smaller size. So we thought, okay, maybe this is how rapamycin is preventing diet-induced obesity in these mice. We are eliciting this transcription factor and thus activating lipid oxidation and engaging this metabolic rewiring described by the Calcoven group. Oops, I went too fast. So we looked at the expression of LAP and LIP in the liver of the mice that I showed you before, and while in the low-fat diet-fed mice, the results were very consistent with the hypothesis, where the lap over lip ratio was clearly increased. In terms of the high-fat diet, actually, we did not see that. But if you look down at the actual plot down here, you'll see that the mice fed a high-fat diet and treated with rapamycin actually had higher levels of both lap and lip of all the groups, of all the 4 groups. So we decided to not just throw in the towel just yet, and look a little bit further. We found a gene that is a reliable reporter of lap activation, and this is this EDOL3. So we looked at the expression of EDOL3 in the liver of these animals, and as a matter of fact, all the mice treated with rapamycin, whether they are on a high-fat diet or on a low-fat diet, had higher expression of EDOL3 via Western blot, which is consistent with the idea that we are maybe engaging the CBP beta mechanism. So we thought, okay, let's try and see if we can confirm this. Is there a way of activating CBP beta independently of mTOR inhibition, and are we going to achieve the same effect as giving rapamycin? And here the Calcoven group came to our aid again, because they had published this in vitro screen where they were looking for drugs that would affect the lap to lip ratio. And one of the major hits, actually the highest hit, was this drug, adefovir, the Pavoxel, which here is drug number 4 in this Western blot, was also able to reduce the levels of lip in vitro, thus increasing the lap over lip ratio, or reduce the lip to lap ratio as it is in this graph over here. And adefovir Pavoxel is another FDA-approved drug, just like rapamycin. It is a drug that is approved for the treatment of chronic hepatitis B. It looks like this, so it's kind of like a nucleoside analog. What we did is we took adefovir Pavoxel, and we asked, okay, does adefovir Pavoxel prevent diet-induced obesity? So we took female mice this time, and we did that mostly because we had a stronger effect of rapamycin in female mice than in male mice, which was to be expected, given what we know about rapamycin, its effect on longevity in a way. And we ran a nice dose response, both via intraperitoneal injection and by mixing the drug in the food. And we asked, okay, can we prevent diet-induced obesity using this drug? And we had a positive control group with rapamycin, and then our untreated groups. And especially when we look, if you look at the food treatment, so with the drug mixed in the food, there's a very nice dose response looking at the % weight gain, with the mice gaining less and less weight on a high-fat diet, the higher dose of adefovir Pavoxel they were receiving. With intraperitoneal injection, you can achieve a better, higher serum levels of the drug. So our dose response curve was not quite a curve, but pretty much all the treatments that we gave were quite effective at reducing weight gain in these mice. So we decided to concentrate our efforts on 2 concentrations. This one over here, the highest concentration in the food, the highest dose in the food, which was 210 milligram per kilogram mixed in food, and this is 10 milligram per kilogram via intraperitoneal injection. And we looked via qMRI at the accumulation of fat. Again, both doses were pretty effective, with the IP injections were a lot more effective at reducing adiposity in these mice. So we decided, okay, let's fill in the blanks and run all the proper controls. So we had another group, another cohort of mice. We had high-fat diet, low-fat diet, and they were either on vehicle injections of dapavir, normal chow, or chow with a dapavoxyl. I'm going to talk mostly about the 10 mg per kg intraperitoneal data here, but the data with the food are pretty similar, just not as striking. And if you pull all the results from both the cohort 2 and cohort 3, again, in percent weight gain, you can see that dapavoxyl blunts the weight accumulation that these mice get with the high-fat diet feeding. You can reduce adiposity as well. And these results are very much driven by actual accumulation of fat in the fat depots. We looked at the weight of different organs at the moment after sacrificing the mice for necropsies. And again, what we saw a major difference was in the epididymal fat pads, in the periproductive fat pads, where we have a much higher mass in the mice fed a high-fat diet, and the effect was clearly blunted, severely blunted by a dapavoxyl. And this was despite slight hyperphagia that these mice were displaying at least 3 out of the 6 weeks of our treatment. So were we able to see the same biochemical changes in these mice that as the Kalkovin group saw in vitro with the dapavoxyl and in the delta UORF mice? Well, again, we can see that the dapavoxyl changes increase the lab-to-lip ratio in the liver of these mice. This is the Western blot. And again, we have confirmed that this is not mediated by inhibition of mTOR, because if you give rapamycin, in the mice fed a high-fat diet, you don't have any signal, any phosphorylation of the downstream effector of mTOR signaling, which is ribosomal protein S6. Or else, if you look at the mice with the dapavoxyl, phosphorylation of ribosomal protein S6 is pretty much unaffected. So how is a dapavoxyl activating CPP beta? This is still kind of a mystery, and this is where we are focusing our efforts now. And as I mentioned before, this drug is a nucleoside analog reverse transcriptase inhibitors. Its mechanism of action is by inhibiting the viral reverse transcriptase. One of the side effects of this class of drugs is inhibition of polymerase gamma, which is the mitochondrial DNA polymerase. So mitochondrial toxicity is sometimes a concern. So we are working under a framework that this drug might be inducing a mitohormesis phenotype in these mice, and the mitohormetic response might be responsible for the resistance to high-fat feeding. We have a few data consistent with that. I'm going to show you just something real quick. And therefore, the dapavoxyl induces phosphorylation of P38 stress-activated protein kinase, which is a MAP kinase that is activated in response to stress. This is the Western blot here. This is just a densitometry. And so we are working along this hypothesis, both using mouse tissues and now with the in vitro and ex vivo models. And hopefully we can confirm this hypothesis, or we will be able to figure out the mechanism of action. But it will be interesting to see if this hypothesis is right, because mitohormesis is a relatively conserved longevity mechanism. So if it can apply to obesity, then we can see how the parallel between the two conditions is actually much stronger than we initially thought. So I'm going to leave you with this idea. I hope I could show you how the knowledge that we've gained into aging biology can actually be exploited to treat and study other diseases that are at least superficially age-independent, like obesity. And it remains an interesting question to determine whether the reverse is also true, if like studying obesity or mitochondrial disease and other age-independent pathologies using a geroscience framework can actually give us new insights into aging biology. And I'm going to stop here, and I'm going to be ready to answer your questions, should you have any. But this is my acknowledgement slide, and I want to thank specifically Matt Kibeline, who keeps me around after all these years, and a number of other postdoctoral fellows and graduate students in the lab who are helping with various experiments. Mitch, who I am trying to convince to run a fly lifespan with a dapavoxyl, and in particular my undergraduate assistants, which I call the Bench Street Boys, who have run most of the experiments that you have seen. This is a cohort that has graduated this past summer, and over here, these are my new students now. some older students that have graduated have gone to better and greater things, and collaborators and staff of the animal facility and previous funding, of course.
All right, thank you, sir. We're out of time for questions, but yeah, if you do have any questions for Alessandro, feel free to add them into the Slack or into the Q&A. He'll be looking at them and he can respond to them as you're adding them.